So I want you to imagine having the perfect, just absolutely flawless blueprint to build this magnificent house.
Right.
You read the schematics and everything looks structurally sound. The dimensions are perfect. But, when you actually show up to the construction site, the roof is caving in, the walls are rotting, and I don't know, the plumbing is totally backwards.
A total disaster.
Exactly. And that disconnect, that gap between what the plan says and what is actually happening on the ground, that is exactly what happens when doctors look solely at your DNA to figure out why your tissues are sick.
Yeah, the blueprint doesn't tell you if the roof is actively leaking right now.
Exactly. To fix the house, you can't just look at the paper. You have to look at the actual building materials that are currently on the site. You need to look at the proteins. So, welcome to the deep dive.
Glad to be here.
Today, we are embarking on a mission, really, to explore a monumental 2026 Nature paper that fundamentally redraws the map of human biology. And I mean that quite literally.
Yeah, it's a completely spatially resolved, quantitative map of the human proteome.
Right, across, 58 healthy tissue types, 25 distinct carcinomas or cancers, and 22 fetal tissue types. Okay, let's unpack this. Why are proteins the missing piece of the puzzle here? Why focus on them now?
Well, it is the absolute definition of diagnostic muddy waters. But your blueprint analogy really hits on the exact paradigm shift this research represents, because for decades, biology has been largely obsessed with the blueprint.
The DNA.
Right. We mapped the human genome. Then we mapped mRNA, which you can think of as the photocopy of the blueprint being sent from the architect's office down to the construction site.
Okay, the instructions are being sent down.
Exactly. But here is the biological reality of our cells, which is wild. mRNA abundance only moderately correlates with actual protein expression.
Wait, really? I always thought, if you have a lot of the RNA instructions, you automatically just get a lot of the protein.
No, it's a super common misconception, actually. But it's not a one-to-one ratio at all. The relationship is highly regulated and incredibly complex.
How so?
Well, you can have a massive amount of mRNA, right? But if the cell's machinery is pausing the translation process, or say if the resulting proteins are degrading rapidly once they're made, the actual functional amount of protein is very low.
Oh, wow. So the instructions are there, but the workers just aren't building it?
Exactly. And proteins are the main functional and druggable molecules in our bodies. I mean, they are the enzymes digesting your food, the structural pillars holding your cells together, the signaling messengers telling your heart to beat.
So they do everything.
They do. If you want to truly understand human biology and disease, especially something as complex as cancer, you absolutely must map the proteins as they actually exist in the tissue.
That makes a lot of sense. And I'm sure some of you listening are thinking the exact same thing right now. Haven't we already done this? I distinctly remember hearing about the Human Protein Atlas years ago.
Yeah, back in 2005.
Right, 2005. So what is actually new here? We've had maps before, right?
We have. And the 2005 Human Protein Atlas was a massive pioneering effort. I don't want to downplay it. But to understand why this 2026 paper is such a breakthrough, we have to look at how they built that original map.
Okay, how do they do it?
Historically, mapping proteins relied heavily on a technique called immunohistochemistry.
Immunohistochemistry, that's a mouthful.
It is, yeah. Essentially, scientists used antibodies to find proteins in tissue samples.
Okay, antibodies. I know those from, the immune system, right? They latch onto viruses and stuff. How do scientists use them for mapping?
So the scientific community engineers specific antibodies that are designed to recognize and bind to one very specific target protein.
Okay, like a lock and key.
Exactly. And they attach a little fluorescent tag or color-changing enzyme to that antibody. Then they wash it over a slice of tissue and look under a microscope.
And so wherever it glows...
Wherever you see a glow or a color stain, you know your protein is there.
That sounds pretty effective, actually.
Well, it is great for getting localized information. You can see roughly where a protein is inside a specific cell. But it has massive, massive limitations.
And limitations like what?
First off, it is only semi-quantitative.
Meaning?
Meaning it gives you a visual estimate of, more or less protein based on how bright the stain is. It doesn't give you an exact molecular count.
Oh, I see. So it's like saying a room is bright versus telling me exactly how many light bulbs are in there.
Exactly. And second, and this is the bigger issue, you are completely limited by whether an effective, highly specific antibody even exists for the exact protein you want to study.
Wait, we don't have antibodies for all of them?
Not even close. Out of the tens of thousands of proteins in the human body, we don't have perfect antibodies for all of them. So if you don't have the antibody, that protein is completely invisible to you.
You know, I was thinking about this methodology, and it's like, imagine you are trying to count the population of a massive city, right? But your only tool is a team of spotters looking for people wearing red hats.
That's a good way to put it.
Right, so if someone has a red hat, which represents your antibody, your spotters see them perfectly. But you completely miss the thousands of people wearing blue hats, green hats, or no hats at all.
Yeah.
You get this wildly skewed, totally incomplete picture of the city's true population.
That is a perfect way to visualize it. You only see what you already know how to look for. But the scientists in this 2026 paper, they ditched the antibodies entirely.
No more red hats.
No more red hats. They shifted to an unbiased, multiplexed approach using mass spectrometry. To borrow your analogy, that is like swapping out your red hat spotters for a thermal imaging satellite.
Oh, wow.
Yeah, a satellite that simply registers every single human heat signature in the city, completely, regardless of what they're wearing. It doesn't rely on pre-existing antibodies at all.
Okay, let's take a quick pause here. For you listening, we're at about the 5-minute mark, and I want to do a quick recap to just lock this knowledge in. So, bullet 1, DNA and mRNA are just blueprints. Proteins are the materials that do the actual work.
Right.
Bullet 2, older protein maps used antibodies, which were incomplete and semi-quantitative, like only looking for red hats. And bullet 3, we need a new tool to see the full picture, which brings us to the tech breakthrough of this paper. But before we get into the satellite part, I have a question for you listening. Can you guess how much pressure it takes to squeeze the biological secrets out of a solid piece of bone?
It's a lot.
Yeah, so building on that recap, let's talk about the insane technological mechanism they used to actually build this new map. Because if I'm tracking this right, before they can even put the tissue into this mass spectrometry satellite machine, they have to get the proteins out of the cells first.
Right, the sample preparation.
And they aren't just dealing with soft liquids like blood. They are analyzing cartilage, solid tumor biopsies, and even solid human bone. Like if I asked you to extract intact proteins from a solid human bone, how would you even start?
It's notoriously difficult.
Right, because these scientists decided to subject the tissue to an environment that sounds like literal science fiction.
You're referring to the pressure cycling, and yes, it is extreme. What's fascinating here is that to extract the proteins, they use a physical sample preparation technique called PCT.
PCT, what's that?
Pressure cycling technology. They subject these incredibly dense tissues to 20,000 psi.
20,000 pounds per square inch. Just for context for you listening, atmospheric pressure at sea level is about 14.7 psi.
Yeah.
20,000 psi is more pressure than if you were sitting on the ocean floor at the very bottom of the Mariana Trench.
It's immense.
Why on earth do they need to subject human tissue to that kind of extreme environment? I originally thought it was like a microscopic pressure cooker, just boiling the cells alive.
Well, it's actually not about heat at all. It's entirely about mechanical force.
Okay, so no heat.
Right. A pressure cooker uses heat and trapped steam. PCT uses immense hydrostatic pressure at strictly controlled temperatures. A better analogy would be a deep-sea submarine.
Oh, I like submarines.
Imagine a submarine rapidly diving to the bottom of the ocean and then instantly teleporting back to the surface.
It would explode.
Exactly. The pressure outside the hull changes so violently and rapidly that the structural integrity of the hull fails and it pops.
Oh, wow. Okay, so the cell membrane is the hull of the submarine.
Precisely. In PCT, they hit the tissue sample with that 20,000 psi for 50 seconds and then rapidly drop it back down to ambient pressure for just 10 seconds.
So they cycle it.
Yes, they cycle this rhythmically. 50 seconds of crushing deep-sea pressure, 10 seconds of surface pressure. This rapid cycling creates intense mechanical shear stress and what we call cavitation bubbles.
Cavitation bubbles, like tiny bubbles popping.
Yes, and that force physically rips open the tough outer membranes of the bone and cartilage cells. But here is the real genius of it.
What's that?
While the large, rigid cell membranes are shattered by the pressure changes, the individual proteins inside are so incredibly tiny and structurally flexible that they survive the decompression completely intact.
That is absolutely wild. They are basically rhythmically crushing and releasing the tissue to pop the cells like tiny balloons, leaving this protein soup perfectly preserved.
That's exactly what it is, a protein soup.
So once they have this liquid soup of extracted proteins, they feed it into the thermal satellite you mentioned earlier, the mass spectrometer. The paper calls it DIAMS. What exactly is the DIA part?
It stands for Data Independent Acquisition. And to understand why it's revolutionary, you need to know how traditional mass spectrometry works.
Okay, Lay it on me.
A mass spectrometer essentially weighs molecules. It ionizes the peptides, which are the chopped-up building blocks of the proteins, and it measures their mass-to-charge ratio in a magnetic field.
Right.
But historically, these machines operated in a data-dependent mode.
Meaning they depended on something specific to take the measurement.
Exactly. The machine would look at the complex sample, find the most abundant, loudest chemical signals, and isolate only those for detailed analysis.
Oh, so it just ignored the quiet stuff.
It essentially ignored the quieter signals, yeah. It was totally biased toward abundance. But DIAMS changes the game entirely.
The data-independent one.
Right. It captures data for all detectable peptides within a specific mass range simultaneously. It takes everything in. It doesn't discriminate based on whether a protein is shouting at the top of its lungs or just whispering in the background.
It captures the whispers and the shouts. That's incredible. And the scale of what they accomplish with this combination, the deep-sea submarine cell popper and the whisper-capturing mass spec, it is staggering.
The dataset is massive.
Let's look at the numbers they pulled off here. They analyzed 2,856 individual samples spanning fetal development, healthy adult tissues, and tumor states. From those thousands of samples, they quantified 13,609 distinct proteins.
And they did it with a false discovery rate of just 0.1% at the protein level.
Okay, but this brings up a massive logistical problem. 13,609 proteins across nearly 3,000 samples. The data matrix for that must look like absolute chaos.
Oh, it's completely unreadable to a person.
Right. So how does a human brain take a spreadsheet with tens of millions of data points and actually turn it into a map that makes biological sense?
Well, the short answer is a human brain can't. This is where computational biology and machine learning step in. The researchers utilized an algorithm called TSNE.
TSNE.
Yeah, that stands for T-distributed stochastic neighbor embedding.
Yeah, I'm going to need you to break down what that actually means mechanically. How does an algorithm map a 13,000-dimensional spreadsheet?
Sure, let's visualize it. Imagine you want to map animals based on their traits, okay? Weight, height, speed, diet, and lifespan.
Okay, at 5 traits.
That's a 5-dimensional data set. Now, we can't draw a 5-dimensional graph. Our brains can't comprehend that. What TSNE does is calculate the mathematical probability that two animals are similar across all those traits in high-dimensional space.
So like finding hidden patterns.
Exactly. A dog and a wolf have a very high probability of being similar. A dog and a jellyfish have a near-zero probability. The algorithm takes those complex similarity probabilities and basically squashes them down, translating them into simple 2D distances on a flat screen.
So it groups things that share the same building materials.
Exactly. The algorithm plots a point for every tissue sample. Tissues that have similar protein makeups are pulled closely together into tight clusters on this 2D map, and tissues with very different protein makeups are pushed far apart.
And predictably, when they ran this algorithm on the human body, tissues with related physiological functions grouped beautifully together.
They did.
Like the brain, the spinal cord, and peripheral nerves all formed this distinct, tight cluster on the map. Because functionally, they're all part of the nervous system, so their protein building blocks are highly similar.
It confirms the biology we already knew.
Right, but here's where it gets really interesting. The algorithm threw out some clusters that, at first glance, look like massive, embarrassing errors, just anomalies that totally blow your mind.
Yes, but the anomalies are actually the most revealing part of the entire study.
Because they prove it works.
Because they prove the map is measuring functional reality, not just anatomical names. The best example of this is the mammary gland.
Oh, this one was crazy.
When the algorithm mapped the mammary gland tissue, it didn't cluster it with other soft glandular tissues. It clustered it tightly with connective tissues, things like solid bone, tendon, and cartilage.
Anatomically, that sounds completely wrong. A breast is not a tendon. Why on earth would the proteins look identical?
It comes down to a biological mechanism called age-related involution.
Involution.
Yes. The tissues sampled in this study were from adults. As a mammary gland ages, particularly after the reproductive years, the specific structures that produce milk, which are called the alveoli, they undergo apoptosis.
That's programmed cell death, right?
Right, programmed cell death. So they begin to decrease and shrink. The tissue actively remodels itself. To fill the empty space left by the dying alveoli, the body deploys adipocytes, or fat cells, and synthesizes large amounts of collagen and extracellular matrix.
So the tissue is physically transforming over time.
Precisely. The proteome is reflecting the current functional state of the tissue. And the scientists didn't just blindly trust the machine learning algorithm either.
They checked its work.
They performed a rigorous physical validation using a gold standard pathological test called H&E staining.
What does H&E stand for?
Hematoxylin and eosin. It's a chemical stain used on tissue slices.
Okay, how does it work?
Well, hematoxylin is a basic dye that stains acidic structures, like the DNA in the cell nucleus, a dark purplish blue.
Blue for DNA, got it.
And eosin is an acidic dye that stains basic structures, like the extracellular connective matrix and cytoplasm, a bright pink.
So they slice the tissue, dye it, and look at the colors under the microscope.
Right. And when they looked at the adult mammary gland tissue with the H&E stain, it visually confirmed exactly what the algorithm predicted.
What did it look like?
There was a striking paucity, basically a severe lack of the blue-stained glandular alveoli cells. Instead, the slide was absolutely dominated by the bright pink stain of the extracellular connective matrix.
Wow, the proteins do not lie. The tissue was functionally, on a chemical level, more similar to a tendon than to a young, active, milk-producing mammary gland. And there was another one that caught my eye, the trachea, your windpipe. It clustered right next to the salivary gland.
Yeah. Again, at first glance, a respiratory tube and a digestive gland seem totally unrelated.
Right.
But if we connect this to the cellular level, we find the mechanism. The specific region of the trachea they sampled has a tremendously high presence of glandular epithelial cells.
What do those do?
These are cells highly specialized for secretion. In the trachea, they secrete mucus to trap dust and pathogens.
Oh, and the salivary gland.
It's also completely packed with secretory epithelial cells producing saliva. The algorithm recognized that their protein building blocks were fundamentally doing the exact same molecular job.
Okay, this is a great time for our second mini recap. We are about 10 minutes in, so if I'm pulling all these threads together, bullet one, the combination of those deep-sea pressure-cycling cell poppers and the unbiased mass spectrometry allowed scientists to capture over 13,000 proteins flawlessly. Bullet two. When they used that TSNE algorithm to organize the data, it ignored the names we gave our organs and grouped the tissues by their actual functional reality.
You have the functional signatures.
And bullet three, this reveals incredible dynamic shifts in our bodies, like the aging mammary gland morphing into connective tissue.
That is an excellent synthesis of the methodology.
Yeah.
But, this raises the most important question of all.
Which is,
why does this matter to the listener? It is fascinating biology, sure, but how does knowing the protein clustering of a trachea or an involuting mammary gland impact human health or potentially save a life?
Exactly. Why should you, listening to this right now, care? And the answer lies in what this map allows us to do next, particularly in the realm of pharmacology and cancer treatment. It all comes down to a critical medical concept called organ-specific toxicity.
Right. Let's explore that mechanism. Because in modern drug development, the biggest hurdle isn't usually finding a chemical that kills cancer cells.
We can kill cancer cells in a Petri dish all day long.
Exactly. We have plenty of chemicals that kill cancer. The hurdle is finding a chemical that kills cancer cells without killing the patient in the process.
Right.
This new proteomic map identified 1,717 proteins that are highly enriched in specific tissues. And impressively, 480 of those tissue-specific proteins were completely uncharacterized before this study.
Okay, so imagine a pharmaceutical company designs a brilliant new drug. It is a highly targeted therapy designed to seek out and attack a very specific protein that is driving the growth of a lung tumor.
Okay.
In the past, they might put this drug into clinical trials and suddenly find that patients are experiencing severe, unexpected heart failure. Why? Because it turns out that same exact protein target they thought was only in the lung tumor also happens to be a critical structural component in the healthy heart muscle.
And this atlas acts as a toxicological GPS to prevent exactly that catastrophic scenario.
A toxicological GPS. I like that.
Yeah, before a drug ever enters a human trial, scientists can now consult this comprehensive, highly specific proteomic map. They can say, okay, our target protein is highly expressed in this lung carcinoma. Where else does it live?
So they just look it up on the map.
Right. If the map shows that protein is also heavily enriched in the healthy brain and the kidneys, they immediately know they have a high risk of severe off-target side effects. They can predict and avoid organ-specific toxicity before a single patient is exposed to the drug.
It is essentially a cheat code for drug safety. But the researchers didn't stop at just analyzing healthy tissue. The paper also directly compared the proteomes of 25 distinct human carcinomas with their healthy, non-tumor tissue counterparts.
They did.
They mapped the cancer.
And the numbers they generated from that direct comparison are the ultimate payoff of this entire endeavor. By cross-referencing this massive data set, the study successfully identified 41 tumor-enriched proteins.
Just to be clear on the mechanism here, when you say tumor-enriched, you mean these are proteins that are highly abundant in the cancer cells, but have practically zero presence in the healthy vital organs.
Correct. They are practically non-existent in the tissues you need to survive.
41. That is 41 incredibly clean, highly specific targets for new therapies.
Yeah, if we connect this to the bigger picture, this is the holy grail of oncology. It allows researchers to prioritize therapeutic targets with a degree of confidence we simply have never had before.
Because we know what's safe.
Exactly. Furthermore, it opens the door for finding repurposable anti-cancer drug candidates.
Repurposable meaning like we already have the drug.
Yes. If a drug already exists that safely targets one of these 41 proteins for a completely different, non-cancerous disease, we now have the hard data to suggest it could be rapidly repurposed to fight cancer.
Oh, wow.
Because thanks to this map, we know exactly where that drug will act in the human body, and more importantly, where it won't.
So what does this all mean for us? We started this deep dive talking about blueprints and building materials. The genome was the blueprint. But this 2026 Nature paper proves that we have finally truly walked onto the construction site.
We really have.
By popping cells under extreme pressure and sweeping them with unbiased mass spectrometry, scientists have categorized every beam, every nail, and every worker across fetal development, healthy adulthood, and cancerous pathology. We are looking at the foundational first step in creating a true, interactive digital navigator for the human body.
It is a phenomenal historic achievement in biology.
Yeah.
But, I want to leave you with a thought to mull over, something the paper hints at but leaves open.
What's that?
This map proves that a proteome is not static. It changes drastically from when you were a developing fetus to a mature adult. And it actively morphs during aging, just like we saw with the physical transformation of the mammary gland.
Right, it's constantly changing.
So considering that incredible fluidity, how much does your personal proteome shift day to day? Are the proteins in your body reacting, upregulating, and physically restructuring right now at this very second based on your current stress levels, what you ate for breakfast, or the environmental toxins in the room you were sitting in?
That is, yeah, it really makes you wonder what your own personal digital navigator would look like at this exact moment. Well, thank you for joining us on this exploration of the human proteome. Keep questioning the blueprint, and we'll catch you on the next deep dive.